So thank you very much, and I thank also Mustak for holding the presentation earlier than me. I was in another CAS seasonal school in Memristor, so I was chairing the session, and I had to leave just at 6 o'clock. So I am very honored to be here and for this very important event to the memory of Professor Tamás Roska. And I'm sorry that I cannot be there in person, and I'm very happy to see also all our old friends, both online and in present. So I hope that there will be an occasion that we all together once again to have another meeting of this kind to the memory of Professor Roska. So since we have a limited time, I will proceed with my talk. That is it has a very long title. It's a long work that we have developed together with the colleagues in my university here in Politecnico di Torino, and also in Technische Universität Dresden. So here is the acknowledgement slide. So I would like to thank Professor Teslav, Professor Chua, some postdoc in Dresden, Demir Kol and Yanis Messaris, and then my colleagues in Torino, Professor Corinto and Professor Gilli. And we have also the experimental group from NAMLab in Dresden. Professor Mikolajczyk is the director and also professor at the University of Dresden. So I tried to make the presentation compact. Sorry for this issue with the animation. But so in this first slide, I would like just to give a brief reminder about the model of the Hodgkin and Huxley that was developed by the two American luminaries, Hodgkin and Huxley, in 1952. And it is considered a bioplausible model for neuronal cells. It is a very complex model, however, and therefore it is not so much used in terms of hardware implementation these days. People prefer, of course, to use a much simpler model in order to have networks with a large number of neurons to make computation in a very efficient form. However, in terms of modeling biology, this model is of course more realistic. And as you can see, it's a fourth order model, so we don't have to go to the details. So these are the equations of the model. Once again, there is some issue with the animation, sorry. And okay, if you give the parameters the proper values according to the paper, and you insert an appropriate value for the input current, you may develop the waveform of the famous waveform of the action potential. As you can see here, the membrane capacitance voltage can have the shape of an action potential. But most importantly is this bifurcation diagram, which is actually at the basis of our work. So this is a very classical bifurcation diagram where you may see how the capacitance voltage, the steady states actually, both the stable and unstable steady states for the membrane capacitance as a function of the input synaptic current. Okay, so there are three main bifurcations that mark the life cycle of an action potential, and they are indicated by the arrows. So as the input current decreases from 180 microampere, as you can see, there is a blue-green curve, which is the DC characteristic of the neuronal cell. The blue part is the stable part. So it means that the neuron is in equation state, in a stable operating point, sitting silent there. But when you see the green line, it means that the equation state is unstable. So there are unstable operating points. And of course, at the first arrow, indicated here by my pointer, there is an off supercritical bifurcation. So there is the birth of infinitesimally small limit cycle oscillations around the capacitance. This is indicated in red, where you have the maxima and minima of the oscillations for each current value. And as you can see, as the current decreases, the amplitude of the limit cycle increases, and at some point, there is another bifurcation here, indicated by the second arrow, which is the subcritical bifurcation, which is the other type of off bifurcation where there is the interesting complementary phenomenon where the operating point returns to be stable, and at the same time, there is the birth of an unstable oscillation, indicated here in brown. You see the maxima and minima of the oscillation. The oscillation has a very sudden increase in amplitude for a brief, small decrease of the current. However, it is important, this window, because this is the bistability regime of the neuron. So in the sense that there is still the global oscillation, the global the stable oscillation that was born with off supercritical bifurcation, now very large amplitude. And then there is, however, also the operating point, which is stable. So depending on the initial condition, the neuron can either be silent or spike. Okay, so firing spikes here. So this unstable oscillation is very important, but at some point you see that the unstable oscillation grows in amplitude and merges gently with the stable one. And again, we have another bifurcation. In this case, it's global phenomenon. It's the so-called global saddle node bifurcation, where the two cycles coalesce together and triggering the all-to-known effect, the famous all-to-known effect. In fact, directly after this bifurcation, the neuron returns to a quiescent point. So this is a very complex phenomena. And interestingly, Leon Chua has demonstrated in 2012 that basically when the neuron undergoes the two local bifurcations, two off bifurcation, is poised on the edge of chaos. So basically complex phenomena, and this is only one example, probably one of the most important examples. Complex phenomena emerge, so the bifurcations that give rise to complex phenomena typically appear when a physical system is on the edge of chaos. Of course, now we don't have time to go through the details of edge of chaos and what it means on the theory, but this is a key point. So what we did, so basically these are just a sketch of the phenomena that are underlie the all-to-known effect. So what happens is basically that the unstable orbit that it was born out of a subcritical off bifurcation when the neuron was on the edge of chaos, grows very, very fast when the current is decreased, and at some point it merges with a stable one in a global phenomenon called saddle node limit cycle bifurcation. And all these three bifurcations, they appear without, of course, changes in the parameters of the neuron, according to the Hodgkin and Huxley model, but just by changing the bifurcation parameter of the current, okay? And you can read that we did also some analysis of the Hodgkin and Huxley neuron model in this paper, and we showed how the sodium ion channel has a fundamental role in the development of this all-to-known effect in the Hodgkin and Huxley neuron model. But in this presentation, we would like to show something else. We would like to show the design of a memristive cell, which undergoes the same three bifurcation cascade as the Hodgkin and Huxley neuron, while using only two dynamical states. So we reduce the complexity, because the Hodgkin and Huxley neuron model is a fourth order model. Now we are using only a second order cell to replicate qualitatively the three bifurcation cascade without changing any parameter in the cell, just by sweeping the input. So we are exploiting to achieve this the rich dynamics of a locally active Memristor fabricated at NAMLab, the company whose director is one of our partners, Professor Mikolajczyk. So the NAMLab Memristor, here you see a packaged volatile Memristor device is fabricated at NAMLab with an area of 2 micron times 2 micron. They are based on niobium oxide, but here is the demonstration that actually this Memristor can amplify small signal when biased under a suitable operating point. So as you can see, this is the DC characteristic of the Memristor on the plot A. If we insert a current, a bias current, and we bias the device in a point P where the slope is negative, okay? Slope negative means that the device has a negative differential resistance. So then we can exploit the local activity of the device. The device can actually generate local energy, an infinitesimal quantum of energy. In fact, if we superimpose on the bias current a small signal current, we are able to demonstrate that basically, as you can see, the local power integrated over time is a monotonically decreasing function of time. So this is the demonstration of the local amplification capability of the Memristor. And we are currently, with Professor Gilli and Professor Corinto, we are actually developing more understanding now about this amplification phenomenon. So in the past, we also used this device for reproducing bifurcation, complex bifurcation in biology through networks, through analog computing electronic networks with fewer dynamical states than the original biological counterparts. Just to give a couple of examples here, we demonstrated how a fourth order cellular neural network employing two locally active non-lab devices is able to reproduce a complex phenomenon observed by Stephen Smale in an eighth order system from cell biology. The idea is basically the following. So he was studying a cell that was basically silent on its own, irrespective of the initial condition. And our cell, so 1/2 of this network, so this is the cell, and then the other copy is here. They are identical and they are coupled by a resistor. So each cell basically is silent on its own. Irrespective of the capacitance initial state and the memristor initial state, the cell is always silent, as you can see here on the left picture, where we plot the memristor state. Okay, so the cell on its own is silent. And what you expect when you couple two cells that are identical and they're coupled by a positive linear resistor, you expect the dynamics to be further attenuated. And so you would not expect anything else than an homogeneous solution, which means that in this case, we plot the two memristor states of the two cells coupled together for a large resistance value, relatively large. As you can see, both memristor states converge to the same solution as in the uncoupled case. This is the so-called homogeneous solution. This is what we all would expect. But if we decrease the resistance, this was what happened to Smale when he was immersing, he was taking two biological cells and letting them interact via diffusion processes. For certain coupling conditions, he was observing unexpectedly the behavior of pattern formation. I mean, of course, now we have only two cells, so pattern formation has a limited sense, but in the sense that he was observing an inhomogeneous static solution. So this is the so-called Turing pattern in the sense that, and Professor Roska has studied this in a very extensive form, as we know, with Professor Chua in various books. So this is a famous Turing pattern. So there is the behavior of the inhomogeneous solution, which would not be expected in terms of our understanding that two cells that are silent on their own, coupled with in a dissipative environment, would not be expected to support such a complex phenomenon. And by decreasing the resistance even further, one can also observe oscillatory phenomena. And this was the phenomenon observed by Smale when he declared that two silent cells, mathematically dead on their own, they were starting to beat together under dissipative coupling, and this was unexpected. He couldn't believe that was possible. With this work, we actually demonstrated not only we designed the cell, but we demonstrated that this is possible if and only if each of the two cells is on the edge of chaos, which is a particular subdomain of the local activity regime, where a system is not only locally active, so not only can behave as a source of local energy, but is also asymptotically stable around the operating point. Okay, so now about the work that we would like to touch upon now, and we just submitted this paper in a paper, so if anybody of you is interested, we can share the paper. So basically what we did is we used the very same cell that was used in the Smale network, but we performed a deep bifurcation analysis of the cell to understand all the possible type of bifurcation, and we identified under different parameter sweeps. So we always considered one parameter bifurcation analysis, but we swept different parameters. And by doing this type of deep study, we were able to tune the parameters of the network in such a way to actually replicate the bifurcation scenario of the Hodgkin-Huxley model. We also observed much more complex dynamics. So this work is much more extensive, but one of the bifurcation routes we identified is actually the very same that Hodgkin-Huxley neuron can achieve under suitable parameter sweep. So under DC parameter sweep. So this is the Thevenin form of the cell that we also used for the Smale work. And this is the Norton form. So of course they are equivalent. And so what we did is we fixed, I mean, in order to just give the focus point, so the bifurcation analysis that reproduces the Hodgkin-Huxley diagram. So we kept fixed the capacitance. Of course, we have a theory by which we choose the parameters of the memristor and of the capacitance. Key idea is that the memristor must be in the locally active regime. So it must be so when we sweep the voltage, this is the load line analysis, only when the voltage, by fixing the resistance, the voltage enters in the NDO regime, we can have complex phenomena. So only in this area, in this red area. So we fixed the capacitance, we fixed the resistance, we swept the voltage, okay? So similarly, as of course, if you look at the Norton form, it has more a form of it has more a shape of the Hodgkin and Huxley neuron, right? Because it's a current controlled. But okay, it's the two systems are equivalent, so one can study one or the other. And this is the bifurcation diagram actually under voltage sweep. As you can see, this is shown in the other way around as we are showing this in the paper. In the paper, we flipped the axis to make it more closer to the shape of the Hodgkin and Huxley neuron diagram. But nevertheless, we can identify, as you can see, from left to right. The first bifurcation is the supercritical off. Then we have subcritical, the second one, and the third one is the fold limit cycle, fold or saddle node limit cycle bifurcation, the global one. So here is a gallery of phase portraits. This is a zoom-in view where you can see better. The first on the left, it's the of course, the shape are different because, I mean, quantitatively, we cannot expect that our second order simple relaxation oscillator, I mean, everybody's working with this type of oscillator, so it's a standard cell, but nobody has ever identified this bifurcation phenomena inside the cell. So we were able to use, gives also an idea about how important is theory, because through the bifurcation analysis, we were able to tune the parameters in a very conscious and systematic form to achieve this reproduction of complex phenomena. So as you can see on the left side, we have supercritical bifurcation, then a subcritical, and finally a fold cycle. This is a gallery of phase portrait. As you can see, 1.12. We are still below the bifurcation, the first bifurcation, so the neuron is silent. As you can see, we have on the left time waveforms, on the right phase portrait analysis. I mean, this is a second order system, so it's very simple and nice to work on the phase portrait, on the phase plane, and not as simple to do this same work in the fourth order model of Hodgkin and Huxley. Here is the first supercritical off bifurcation. Of course, as you can see, there is the birth of an infinitesimally small limit cycle, and the operating point becomes unstable. So as you can see here, a trajectory initiated far away from the operating point. The blue one goes into the red cycle. Maybe you cannot see this on the left, but on the right you can see it. And but if you start inside the cycle, again, you go into the cycle because the cycle is globally asymptotically stable. I see that I have only 1 minute. Yes, thank you. So I will just finalize, just giving this gallery of phase portrait, and for the explanation of the theory, perhaps we can provide the paper. So this is, of course, the first bifurcation, and the operating point becomes unstable. Then moving on for 1.14, now the global asymptotically stable limit cycle has larger amplitudes, as you can see in the time wave form of the voltage across the capacitance. And this is, as you can see in red, the larger now limit cycle. The two trajectories in blue and black, of course, move to the limit cycle in red. The operating point, as we know, is unstable. And then at some point, there is the subcritical off bifurcation. So now we are at the point where my pointer is moving here. So what happens is that, as you can see, the blue trajectory moves to the red orbit. The black one also moves to the red orbit, but we have another cycle that has just been born in magenta. It's unstable. So the green trajectory actually moves toward the operating point that has returned to be stable now. So we have stable oscillations in red, unstable oscillations in magenta, and this is the trajectory that inside the unstable cycle moves to the operating point. Finally, when we go into the bistability regime, the unstable cycle grows. As you can see, the magenta cycle is much larger, it's getting closer to the red one. And so as you can see, the unstable oscillations are very close to the stable ones. And finally, we have the global phenomenon where the two cycles merge together. You see now, this is a semi-stable oscillation. So everything initiated outside converges to this fold limit cycle. Everything initiated inside converges to the operating point. And after this point, we have the silent phenomena, of course, and the trajectory spans a lot of revolution around the area where the two cycles just collided, but finally, asymptotically, eventually evolves towards the global asymptotically stable operating point. So we have no time to explain why edge of chaos is the origin of these neuronal bifurcations, but the theory is probably the nicest part, but I don't have time to explain it. I would like to thank all the audience, and again, I have a last thought for Professor Tamás Roska that has definitely contributed to my growth over the years, and so I still remember the time where we were also there with Professor Gili, Professor Corinto, and Professor Teslav 10 years ago, and I hope that we can return there and to have another meeting of this kind to the memory of Professor Roska. Thank you.